Can we tell where a biological sample has come from based purely on its DNA? This is what contestants were asked in the CAMDA 2020 Metagenomic Geolocation Challenge. I'd like to walk you through the approach that we took using metagenomic geolocation based on read signatures. We share our bodies and the entire planet with microbes that are critical to our survival. With most of the world's people living in urban areas and many of them using public transport, our transit systems are a microbial melting pot of interactions with human beings. This is why the MetaSub Consortium has sampled from these urban environments, and that leads us to this year's CAMDA Challenge, which asks, does the DNA in these samples reveal information that can identify a sample's city of origin? Can we represent these measurements in ways that preserve information that's informative of location? CAMAs offer a computationally appealing way to represent sequence data. Our motivation in this CAMDA challenge is to explore the extent to which these CAMA representations can usefully preserve information. In essence, there are four main steps to our investigation. First of all, we chose to remove reads that we felt wouldn't tell us very much about particular locations. We removed reads that mapped to the human genome, and we also explored removing reads that map to bacterial genomes that are endemic to human beings. This plot shows the percentage of reads from the samples of each city that aligned to the human genome. Some cities, Offa and Lisbon, had very little in the way of human DNA, whereas others, including Tokyo and Sendai, had quite a lot. The inset here shows the distribution of read lengths with counts on a log scale, showing that merged reads range from about 50 bases long to close to 300. The next step in our analysis was to represent these reads of variable length by CAMAs and encode them as fixed length read signatures. We can think of this as using a lookup table for each of the CAMAs and encoding them using a fixed length vector. Here we've chosen random vectors that are orthogonal to one another. We sum those encoded representations up and normalize to produce a unit vector of the sum of encoded CAMAs to produce what we call a read signature. There are a number of ways in which we can do this encoding. Here we've shown this as a sum of random vectors, but other strategies are possible. After we've converted each variable length read into a fixed length read signature, our task is then to compress these read signatures into a more compact representation that characterizes the location from which the reads came. We actually randomly sampled 1 million read signatures from each location, basically to save time so we could meet submission deadlines. Then we used clustering to find a much smaller number of representative location signatures. We used tree-structured vector quantization, which is an hierarchical k-means approach, and produced clusters or location signatures of size 1, 10 or 100. So graphically, we take these variable length reads, we convert them to read signatures. We use clustering to produce a much smaller set of location signatures. Once we have this smaller set of location signatures, we can then use standard distance metrics to calculate the similarity between these signatures. When we get a novel or mystery sample, all that we need to do is calculate the distances between its location signatures and the signatures of samples that we know. You can use a range of distance metrics, and it's important to explore these, especially given the behaviour of these metrics with high dimensional data. Once we've assessed the distance between the location signatures of a novel sample, we then vote to determine the known city that has the most nearest neighbours to that unknown sample, and that's how we decide on the classification of that unknown sample. We use leave one out cross-validation of the data to see whether we have retained information that's informative of sample origin. And from this confusion matrix, we can see that we've done a pretty good job with many cities in figuring out correctly the city of origin. There are some cities that prove challenging. So, for example, Lisbon here was consistently difficult in all variations of the experiment for us to classify. Very exciting to see this strong diagonal in the confusion matrix. In fact, we were quite surprised to see that 60% of these predictions in this case were correct. It's important to remember that we've used fairly basic methods. Our desire here was to prove a concept rather than to tune performance. And clearly, more sophisticated prediction methods and distance metrics could be applied. What we were trying to explore, though, was whether the CAMDA representation could carry meaningful information about sample origin. We investigated the sorts of factors that would influence the performance of this pipeline and found that performance improved with increasing CAMDA length. We also see increasing performance as we increase the number of location signatures. And pleasingly, we see that our idea that we could remove non-informative reads does actually seem to improve the classification performance here. Still, we think these overall classification rates are surprisingly good given the complexity of the task and the amount of information compression that has been involved. In summary, can CAMDA representations help reveal sample origin? It seems so from the CAMDA data that we've analysed, but there's plenty more work to be done. We hope you've enjoyed this brief tour of the approach that we've taken, and we would welcome questions or ideas from anybody else interested in adopting this approach.